Hello students, warm wishes. In this particular session, we will be discussing the replication of the ends of eukaryotic chromosomes. Ends of eukaryotic chromosomes, we know that it is known by the term telomere, and we will be discussing the role played by the telomerase enzyme for the replication of that end. So, before discussing that telomerase activity at the terminal portion of the eukaryotic chromosome, we have to discuss what exactly mean by the telomere. So, let us see what exactly mean by the telomere. So, telomeres are the unique structure that is found at the ends of eukaryotic chromosomes with tandem repeats of nucleotide sequences. Okay, so they are the unique structure found at the ends of eukaryotic chromosome with the tandem repeats of nucleotide sequences. What is actually mean by the tandem repeats? A particular set of nucleotides are repeated so many times. That is what actually mean by the tandem repeats. For example, a nucleotide sequence TTAGGG, that is in case of the human beings, that will be repeated around 120 to 275 times, and this is what actually mean by the tandem repeats. And this tandem repeats of nucleotide sequences, you can find it here at the terminal portion of the eukaryotic chromosome, is known as the telomere. Actually, this telomere concept was first coined by Hermann J. Muller, and when he was studying the Drosophila chromosome. Okay, Drosophila chromosome in 1938. Okay, so the concept or the term telomere was coined by Hermann J. Muller. Hermann J. Muller in 1938, and he was observing Drosophila chromosome, and at that time he just noted this telomere part. Mainly, this telomere has a definite function in case of the eukaryotic chromosome. So, what are the main functions that is provided by the telomere? So we can first think the prevent DNA's, what is actually mean deoxyribonucleotide, okay, degrading enzyme from degrading the ends of linear DNA molecules. Okay, so it will be actually preventing DNA's activity at the terminal portion of linear DNA molecules, because these linear DNA molecules are more susceptible to DNA's activity. So that will be prevented by this telomere presence. You can find a T loop at the telomere of a human chromosome here. And this T-loop concept is actually beyond the scope of our current discussion, so I'm just leaving it. But a telomere in human beings will be ending like this in a T-loop form. So you can just think about it, and it will be preventing DNAs from degrading the terminal portion. Now the second thing is it actually prevent the fusion of the ends with other DNA molecules, because definitely after each replication there will be a segment of DNA with a single strand form at the terminal portion, and this will be actually pairing with, there is a chance for pairing with another chromosome with a complementary base pairing, and that is actually prevented with the help of the presence of telomere. That is the second function that is provided by the telomere, prevent fusion of the ends with other DNA molecules. And the third function is actually it facilitates the replication of the ends of the linear DNA molecules without the loss of material. So that is the main point that we will be discussing in this particular session. So it is actually facilitating the replication of the ends of linear DNA molecules without loss of material. Otherwise, what will happen? In the absence of this telomere, a terminal portion of the lagging strand will be actually removed after each replication, because this terminal portion will retain as such, because there will not be any free 3-OH at that particular end. As a result, this synthesis will not be taking place, and as a result this terminal portion will be removed. So each replication will be removing this particular terminal sequence. So that means finally it can touch a particular gene of active. Okay, so as a result, it will be damaging the DNA. So that can be prevented by the presence of telomere. So that is the third function. It facilitates replication of the ends of the linear DNA molecules without loss of material. Okay, now this telomere, as I told you, it is actually formed of tandem repeats of nucleotides. So these tandem repeats of nucleotides in eukaryotic DNA terminal portion will have some characteristic pattern. That's what we are going to discuss now. So this telomere basic repeat unit has a pattern in this way. So at the 5' end, it can have a T, and that will be repeated. This T will be repeated 1 to 4 times, and then it will be followed by Na that can be absent or it can be present only one time, and that will be followed by a G which will be repeated 1 to 8 times. So as I just discussed the human TTAGGG, you can see that TT here is repeated 2 times. A, it is only one time that is appearing, and the G, that is repeated 3 times. So and this is actually forming this particular tandem repeat. So this will be repeated many times. This sequence will be repeated many times. So each repeat is having a pattern 213, 2T, 1A and 3G. So that is what happening with the tandem basic repeat units pattern. So we make sure that you are having this idea, because that can appear for CSIR exam. Okay, this is the current general pattern of basic repeat unit that is present in the eukaryotic chromosome. Now, we are just taking some examples that is present in some lower organism and also in higher eukaryotic organisms. For an example, we are just taking an humans, so that's we already discussed the TTAGGG, and in case of a protozoan, okay, Tetrahymena thermophila, the repeating unit is actually TTGGGG. And in case of the Arabidopsis thaliana, it is one of the model organism, model plant species, Arabidopsis thaliana. The tandem repeats are actually having this particular pattern, TTTAGGG. So according to the species, according to the cells within a species, according to the chromosomes in a particular cell, the tandem repeats at the terminal portion will be varying. So that is the main important point. The number of copies of this basic repeat will be varying from species to species, from chromosome to chromosome, within a species, even on the same chromosome in different cell types. So there are two differences you can find. The type of repeat will be varying from species to species or organism to organism. Okay, but you can have more than one organism having the same repeating pattern. Now, the second difference is in how many number of repeats you can find. So that will be different from species to species. It can be varying from chromosome to chromosome within a single cell, and it can be varying of the same chromosome within a species but at the different cell types. So that number will be varying in this pattern. So another important factor that we have to remember. Now, in normal cells, I'm speaking about a non-cancerous cell. So a normal somatic cells. We know that we are having somatic cells and germ cells. And the germ cells are actually moving in from one generation to the next generation. I'm not speaking about the germ cells or any cancerous cells. I'm just speaking about the normal somatic cell. These telomeres usually contain 500 to 3000 repeats and gradually shorten with age. So with age, the number of repeats in a telomere, that will be gradually decreasing. So it is another important concept that is also beyond the scope of the discussion of this particular current session, and we will be having a session separately on how the telomere length and age are related. So we will be discussing in a another particular session. So the telomeres of germ cells and cancer cells will not be shortening with age. So only somatic cell telomere will be varying and shortening with age, and the cancer cell and germline cells will not be undergoing the shortening of telomere with age. Important point, don't forget. And when we are observing different types of telomere, we can find that most of the telomeres terminate with a G-rich single-stranded region of the DNA. G-rich single-stranded region. That means it will be ending with a GGG in that way. And there will not be any complementary base pairing that is happening here. That's what I mean by the G-rich terminal single-stranded portion. DNA strand with a 3' end and are therefore called a 3' overhang. Why? Because it will be appearing like this, complementary base paired terminal portion, and then it will be extending as single one. And this will have a G-rich segment. And this particular portion is known as the 3' overhang. Hope you got it. So this telomere will be ending in a G-rich single-stranded extension at the 3' end, and that will be known as the 3' overhang. Okay, so it will be known as the 3' overhang. Now, this telomere will be repeated how many times? As I told, that will be varying. The number of copies will be varying from species to species. For example, if you are just taking a ciliate, you can find it here. So, if you are just taking a ciliate, it will be repeated 12 to 16 times. Okay, so 12 to 16 times it will be repeated. So, it's not about the repeats. Actually, I mean the terminal sequence. We are just speaking about the overhang, and this overhang segments will be longer and shorter according to the species. Okay, we are speaking about that. Unfortunately, I just went into the previous topic, the number of copies. Okay, we are just speaking about the overhang, and this overhang will have a different number of bases in different species. So if you are just considering a ciliate, that is tetrahymena, you can find that this overhang will be only 12 to 16 bases. That means it will be extending from the double strand from 12 to 16 bases. Okay, so just like 80 in that way. But in case of the human beings, we can find that it will be hanging along 125 to 275 bases. It's too long. If you're comparing with that ciliate, the terminal portion will be too long in case of the human being. So that is also varying in telomere property. Clear? So this overhang will have a different number of bases from species to species. That's why we have given a comparison between a ciliate, that is tetrahymena. It's just like a paramecium like thing, the ciliated animal, protozoan. And we are having human comparison. So it's having 125 to 275 bases long, 3' overhang. In case of the ciliate, it's having only 12 to 16 bases of 3' overhang. Now we are just moving into the role of telomerase. As I told you while discussing the important functions of the telomere, the third function, it's actually facilitating the replication of the terminal portion of the DNA without being uncopied. It's actually allowing the copying of the terminal portion of the DNA. That's one of the functions that we have discussed for the telomere. So we are just discussing now the important topic of today's discussion, that is the replication of the ends of eukaryotic chromosome and how the enzyme telomerase is playing a role in that. So what is the issue with the terminal portion of the eukaryotic chromosome from being copied or from being replicated? So let us see that. So usually the DNA polymerase, this particular enzyme that is responsible for the replication, cannot replicate the terminal DNA segment. The terminal DNA segment, as we have already discussed, the term is telomere. So this terminal DNA segment of the lagging strand, okay, of the linear chromosomes. What is happening with that? You can find it here. So this lagging strand end is, the terminal portion is represented here. And we know that in case of the lagging strand, the synthesis of the new strands, the complementary strand is taking place in this direction, right? So this is the new strand that is being synthesized, and at the terminal portion you can find the RNA primer. And this RNA primer, then it will be extending as the DNA polymerization, DNA deoxynucleotide addition. So after this addition, this Okazaki fragment, this RNA primer will be removed, that we know. Now you can find the gap. No other enzymes can play a role, or the DNA polymerase cannot add DNA nucleotides in this portion where the RNA primer is removed. So a single-stranded extension you can find here. That also should be copied. Otherwise, what will happen if this portion is not copied? This will be removed by the enzymes. As a result, this portion is lost from the parent DNA. Now, in the next time, what will happen? The terminal portion will be here. This will be removed after that replication. Again, this will be removed after that replication. So it will be coming towards the inner portion of the DNA. As a result, sometimes it will touch the gene that is crucial for the life, and as a result, the cell death can happen. So that should be avoided. As a result, this particular segment that is complementary to the RNA primer should be preserved. So for that purpose, we are having a mechanism that is forwarded by the telomerase activity. So here there is no 3-OH. That's why the particular addition of DNA nucleotide is not happening. There is no T-OH. So this gap should be filled, and that is with the help of telomere. That's what this point says. So at the end of the DNA molecule being replicated discontinuously, that's about a lagging strand. There would be no DNA strand. There would be no DNA strand to provide a free 3-OH for polymerization after of the deoxyribonucleotides. Okay, after the RNA primer of the terminal Okazaki fragment has been excised. So there is no free 3-OH. As a result, DNA polymerase cannot add a particular nucleotide sequence there in place of the RNA primer that is being removed. So that is the situation with the end of lagging strand. We have to overcome that situation, and that is done with the help of telomerase. Okay, that is done with the help of telomerase, and let us see how it will be happening. So let us see the mechanism that is played by the telomerase for copying of such single-stranded extension. So let us see that particular thing. So here you can find that the first step in that particular process done by the telomerase is this particular enzyme telomerase. Okay, this particular enzyme telomerase will have a, so let me copy this pen. This particular telomerase enzyme will have an RNA containing, it's an RNA containing enzyme. So it's having an RNA template within this particular enzyme. This is within this particular protein. And that will be providing a neat mechanism for the replication of this telomere at the 3' end of the lagging strand. So please keep in mind that this telomerase is having a built-in RNA primer or RNA template in it that is actually responsible for the copying of this particular segment or extension of this particular segment. Let us see how this process will be taking place. So what will be happening? This telomerase enzyme having an RNA template in it will recognize a G-rich telomere sequence on the 3' overhang. 3' overhang. That means the extension part, extended part, which is single-stranded at the terminal portion of the DNA. So this telomerase will be recognizing a G-rich telomere. We have found that this G will be present in 1 to 8, right? In addition to A, only 0 to 1, T 1 to 4. So who is rich? G only is rich at the telomere portion. So that will be recognized by telomerase enzyme. Then it will be binding with the complementary base pairing using the RNA in it. That is the third stage. With the aid of built-in RNA template, the telomerase extends a telomere that will be binding here. Then it will extend. So now you can have a complementary. You can find here C, 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 A, U. So it's having a template strand here, even though it is RNA. So considering this RNA as a template, DNA extension will be happening at this 3', 5'. So in that way it will be happening. Okay, so this telomere extension will be taking place. So what will happen in this direction? It will be taking place. Okay, so in 5' to 3' direction it will be taking place. So here complementary base pairing will be happening in this way. So U means it will be adding A means it will be adding T, then T, then G, G, G. In that way, in this direction 5' to 3' direction, this particular mechanism, complementary addition of the DNA strand based on the RNA template will be done with the help of telomerase activity. You can find this RNA template that is present within the telomerase enzyme. So this addition will be taking place. So that is the Second step, with the aid of built-in RNA template, telomerase extend telomere in 5' to 3' one repeat at a time. You can find it here, TTA, GGG. So in that way, one repeat at a time, it will be happening. Now again, what will happen? This AAU will be binding now at this TTA. And again, this extension will be happening. That is the third stage. Okay, in third stage, what is happening? This will be repeated several times. So you can find it here. After several telomere repeat units are added by telomerase, the DNA primase add RNA primer to the 3' end of the template. So now you can find here, it will be extending like this, and as a result, you are having large number of repeats of this 3' overhang. So this dotted line represents it is continuous. Okay, it's large number of nucleotides are present in between. Now what will happen at this 3' end, which is actually artificially formed. It is not the original DNA segment part. Instead, it is actually added by the telomerase enzyme. So at this 3' end, the primer will be added, RNA primer will be added. That's what here, by the enzyme DNA primase. Now what you have, it's having a free 3' OH at this portion. It's having a free 3' OH at this portion. Now DNA polymerase can come. This DNA polymerase will be utilizing this free 3' OH at the 3' end of the RNA primer, and this will start adding deoxynucleotides, and it will be extending the chain in this direction. So it will be touching here. Clear? So that is the stage. So this telomerase and the DNA primase add the RNA primer to the 3' end of the template. Now the DNA polymerase catalyzes the polymerization by adding deoxynucleotides to the free 3' OH of the primer, and so on. And so on, in the sense it will be added further. Okay, when you are adding a 3' to the 3' OH of the RNA, a new DNA nucleotide, a DNA nucleotide OH at a 3' end will be exposed. And again, that OH will be utilized for addition of the second nucleotide, and so on, and this will be happening. Okay, and as a result, it will be completing the synthesis of complementary strand till at this point. Okay, now what will happen? The normal process, removal of this primer will be taking place. And this removal will not be creating any problem. Why? Because there will be an extended single stranded portion due to the removal of this particular primer. But this extended single stranded portion is not the original portion of the DNA. Instead, it is the newly added portion of this particular DNA strand, and that is being added by telomerase in this current process. It is not the original DNA part. As a result, if you are removing this segment, there is no issue to the DNA, because the original DNA length is not decreasing by removal of this particular segment. That's what happening with the telomerase. Okay, so that is the important thing in case of the telomerase activity. Now, the significance of telomerase. The significance of telomerase, you can identify what happens in the absence of telomerase. That is actually the significance of telomerase. So, without telomerase activity, what is the issue that you will be facing? The linear chromosome would become progressively shorter, because after each replication, a part of the chromosome that is extended or that is in the form of 3' overhang will be deleted. And finally, it will be moving inner of the DNA, and that can touch actually this particular original recurred gene. That will be lethal. Okay, that's what happening. So that can be prevented by telomerase activity. So without telomerase activity, this can happen. So telomerase activity actually prevents that particular thing. Now, second thing, if the resulting terminal deletions are extended into the essential gene or genes, this chromosome shortening would be lethal. That's what I said. Now, most human somatic cells, most of the human somatic cells lack telomerase activity. Okay, don't think that your somatic cells are having telomerase activity in full-fledged form. Instead, it lacks telomerase activity. That's why somatic cells will not be dividing, or it will undergo senescence or death after some division. Okay, after some times division. That means it can repeat. For example, in case of the culture media, if you are just culturing a somatic cells isolated from your body, it can divide up to 20 or 70, 20 to 70 cell divisions. After that, it undergo death over the senescence. Why? Because it lacks telomerase, and as a result, you are just losing a telomere portion after each cell division. After each cell division means after each replication. And finally, you will be having a least telomere length, which is not effective for the normal functioning of the cell. As a result, the cell undergo death. It will be happening. Senescence will be happening. So we keep in mind, somatic cells lack, most of the somatic cells lack telomerase activity, and that's why it is mortal. It is not immortal because the telomerase activity is not there. It will undergo death after 20 to 70 cell division. Keep in mind that particular thing. But what is the case of cancer cells and germline cells? This is having a full-fledged telomerase activity, and as a result, it is immortal. That's why cancer cells are immortal. It's having a full-fledged telomerase activity. But remember, it is not always the case with all type of cancer cells. Okay, most of the cancer cells will have this telomerase activity. So if you can block the telomerase activity in cancer cells, you can actually block the cancer too. But it's not applicable to all type of cancer because all type of cancer cells are not based on the telomerase activity. So keep that also in mind. And also, in addition to the cancer cell, normal germline cells are also having telomerase activity. That's why it is not undergoing death. It's having acting as the stem cells and all those things. And also it is acting as the cells that is actually generating the gametes. Clear? So it's having a fully fledged telomerase activity. So keep in mind which cell don't have a telomerase activity, which cell don't have a, which cell have a telomerase activity. Keep those points in mind. I just want to wind up this particular session by just mentioning the situation progeria. Okay, so progeria is actually early aging. So within the teenager itself, the person will look like an old age person. So that situation is called progeria. And in case of the progeria, what the scientists can found is, what the scientists found is actually they are having, okay, so very short telomere sequence. So their somatic cells are not regenerative. So after some divisions, that's actually undergoing death. Okay, so they are short telomere. This is one of the characters that is shown by the cells of the persons who is suffering from progeria. And there are other reasons too. For example, WRN mutation. Okay, so that is WRN is actually a protein which is concerned with the DNA repair mechanism. So that is also a factor. It's not always this one, but in case of the progeria, this is also a characteristic feature, short telomere. So if you are having your telomere long, your cells can divide more times. If your cells are having less or short telomere, your cell will not be dividing so many times. Instead, the number of times of the division will be again further shortening. That's what the relation between telomere length and number of cell division that a cell can undergo. So keep those points also in mind. And there is another important factor that the relationship between the telomere length and the aging process in humans. That's another important topic, and that we will be discussing later. So I just wind up this particular session. Hope you understood all the concept very well. And this telomerase activity is most important for the university PG examination as well as for the CSIR UGC NET GRF examination. Okay, thank you for watching. Bye.